Good afternoon. Thanks for staying without break. My name is Sergey. I'm at the Center for Sickle Cell Disease, and I have a variety of activities, but my long-term interest was in proteomics, and so that's actually was the reason why we decided to set up this core, and I proposed it to the RCMI renewal, as a part of the RCMI renewal plan. And I'm going to talk to you about how we think we should structure this new facility, about our aims and our mission. I'll show you typical workflow of proteomic experiment, and discuss where we are now, show you the piece of equipment which we have, and describe our plans. Right, so that's basically what we have now in terms of personnel. It's myself. Actually, I'm trying to write a grant now for Department of Defense. I found a good speak up. Okay. They say I could also be called lab commander, and I kind of like this new title for myself, and I should change to commander. But anyway, so we have. I'm lucky to introduce Dr. Yuri Obukhov. He's a physicist, and we're going to iterate mass spectrometer and basically help us to understand the physics in this process. And also we have Dr. Tanya Amosova, who is a co-investigator, and she is actually a research assistant professor at the Center for Sickle Cell Disease and Department of Medicine, so she can help with the biological part of the sample preparation, talk to the customers or to the users of the facility. So right now we have temporarily allocated our equipment in the hospital. I hope it's not forever over there, but we just never know. So basically we have in this room 3C27, it's Department of Obstetrics and Gynecology. We have most of our equipment, which includes the mass spectrometer. It's LTQ XL Orbitron from Therma, and it can be operated with both electrospray and non-electrospray ionization source. We have non-LC from Shimadzu, and we have also Finnegan micro-LC or HPLC, which also has a alpha sampler, which could be used actually separated from mass spec if someone is interested. And we have a big mining program, Proteomics Discover, which was supplied by Therma and which comes with licenses for Cyclist and Discord, and it could be actually upgraded to other database search programs such as Mascot, for example, if someone is interested. So we, in addition to that, we have actually a bunch of equipment, which most of it is in my lab on 6B24 in the hospital, actually close to the proteomics core, and it includes imaging station, which is a very expensive piece of software which allows us to analyze 2D protein gels. And we also upgrade this imaging station to be operated in the automatic mode. So we have a couple of bioanalyzers which can be used by the users, and it includes a fax machine, which I guess is earlier. But not only, but I have a long list of people who are interested to work with this fax, because it's very simple, really easy to use fax machine. We have another HPLC system, a purifier, and we have light cycle, which is I think the only one, this kind of real-time PCR machine covered, and it could be used for real-time PCR, some of the interested, but also can be used for proteomics, because people say that you can use it for the protein pressure studies using Fred, and it's actually been used this particular application. And also we have full setup for 2D gels, but it's in another room, and we're just waiting for our lab to be finished. And honestly, once it's done, we just move to there. So basically, our mission was to study or to apply mass spectrometry for studies of infections and chronic disease, and we sought to do that by acquiring mass spectrometer and hiring personnel, providing training and support for the Harvard University users and interested parties at Harvard University. And this would actually increase the understanding why mass spec is important, and that's broaden the understanding and multidisciplinary use of this facility, and of course it will enhance the research, and also to bring together actually both basic research, researchers and myself, the clinical research by helping or crafting the analysis of the clinical samples as well. So our original timetable was to year 1 acquire mass spectrometer, hire personnel, undergo training. Then in year 2, which is what we're now, to do the start communicating with user community, start sample collection, start seminar series, and basically teaching course, which people have talked hasn't been in place yet. However, we do have, I think we conduct one seminar and basically in years 3 to 5 they should be fully operational functioning. So that's just an example, as I said in the beginning, how we can arrange community users who could come and work with us, and this would include my group to work on HIV, and we're interested in protein identification and post-translational identifications of proteins, and also focus of Dr. Gurdjieff, the director of our center, who is interested in proteins as they relate to the iron metabolism of hypoxia. We're talking to different community users, I'm not going to name now, but we're interested in protein phosphorylation again and protein signaling, analysis of proteins involved in H. pylori infection and related cancer or hepatic ulcer, proteins involved such as angiotensin II or IGF-1, cardiac hypertrophy, effect of EGF on lymphatic vessels development, proteins involved in breast cancer, and also profiling of different cancers, especially in the African Americans. So these are that's just an example. So everybody can come and can be interested in looking for new proteins or protein modification and then work with this. So as an example, that's the whole study which I did 20 years ago. Now it's about 10 years ago. And the workflow then was actually quite different from what is sought now. So at that time, I had to use large amount of T-cell, and we used about 100 liters of T-cells to generate a lot of cellular material, purify it over several columns, and do like sophisticated fractionations shown here. And then after that, I had to cut out the bands, preserve them basically, and ship to outside facility, which in that case was Harvard microchemistry facility. And we were able to actually find out a few proteins which jumpstart my research, and then I still continue that. But the problem was that first we need a lot of material, second it's very kind of slow process and very expensive, so each line analysis would cost about $10,000 I think. So if you have many samples, basically it's not feasible to do that, and it's very slow at the same time. So you have a long line of people waiting to do the analysis at Harvard, and then if you need to do something quickly to fix your paper, it's not going to work. So, and also at that time I think the non-LC was not in place, so people used liquid chromatography, and then also Finnegan LCQ, which was like a very machine compared to what we have now. So I did a recent study in Russia. This was collaboration between St. Petersburg Technical University, where they acquired very expensive machine, in fact, and they used they bought variant FT transformer. So it's basically it's a machine which has an ion cyclotron analyzer in which ions basically fly around in magnetic field, and you can evaluate their emission spectrum and very accurately estimate the mass of the ions. So in this experiment, the workflow basically was much simpler. So we used cells grown in tissue culture, separate cells on the gradients, then basically immunoprecipitate proteins associated with RNA pol 2, directly then isolate proteins on the SDSP gel, trypsinized, and subject to the MS analysis. or we, after RNA protein column, we did an additional purification step and also did SDSPH trypsin and then MS analysis. And this was actually MALDI-MS. I'm not going to talk about it, but I'll tell you why I don't think it's good for us. So basically, that's the experiment shown here. So because we didn't have much material to begin with, we didn't see many bands, but we did found a few bands which were analyzed, and we, just to make the story short, we did several hits. We found one protein, it's part of the U2 small nuclear protein complex, which had good coverage in terms of sequence coverage, and this was done by mass spot analysis. So, but the backdrop of this whole story with the machine was that we can't do, because it was multi-machine, we can't do MS analysis less successfully. So we end up just doing MS analysis. So we had very accurate mass analysis of the peptides, but at the end of the day, you just don't know what the peptides are dealing with, whether that's true or false. And it also fell into the, I guess, the caveats of the mass spot program, where you end up just putting your life online with this program and say, okay, well, there's a peptides in the program giving back, and whether it's true or not, you just do it. So it tells me basically that we need something different. So when I started to talk to the different companies, I realized that maybe we should go with RCMS rather than multi-MS machine. So I was able to leverage a very good machine, which is Therma LTQ orbit trap. It's basically top of the line machine still, because it's not their top model, but it's still very good model. Very expensive, hard to acquire through the ordinary means basically. So we have as a front line of this machine, we have Nano LC from Shimadzu, which could be connected to nanospray ionization source. And as I mentioned already, we also have regular electrospray ionization source as well, which could be used with the micro-SIM machine. And our mass spectrometer basically consists of two parts. It's a linear trap. So you have ions flying through the ionization source here into this linear trap, where you can analyze ions, sort them, and you can also degrade them right there, and then you can fly them into this second machine, which is orbit trap, which is similar to the FT transformer by Varian, except the size. It's a much smaller machine, so it has kind of a small chamber where the ions fly around, and you also measure basically the frequencies and the convolution, and measure very accurately the mass of the ions. We have several ways to do the MS analysis here. We can do collision-induced dissociation, we can do high-energy dissociation, and also pulse-Q dissociation as well. So, and I'm going to give you some examples. So, because most of the people who first started to do MS analysis, we started from the electrospray ionization, and that's an example of the spectrum here, which we didn't know it in the beginning, but we know now that there is a lot of noise here, which comes probably from the sheet gas, or the gas which helps to evaporate the sample and kind of dry it out up to make the ions, to get the ions, to get the peptides charged from the droplet when the droplets evaporate. So, but nevertheless, you can see signal here. It's a short peptide, and we can the amount of the signal, or the amount of the ions, is proportional to this normalization number, which is 10 to the 5 approximately here. And if we plot the total ion current here, you can see it looks like a chromatogram, because that's how this sample was actually injected into the machine through the small loop, this experiment. And you can isolate this ion and do MS analysis. And that's an example of this analysis. So you can actually, all this information from those ions, you can see that unlike MALDI, ion spray ionization generates multiply charged ions, and this actually makes us much better candidates for the much better and gives you more homogeneous ion population. You can analyze this population of ions through the cyclist, and basically just an example how cyclists look. The cyclist search looks like. So you have your activation or MSMS analysis type. It's collision-induced dissociation, and you'll hit the peptide which we should into the machine. However, I just like to mention that if you try to do that with much less accurate mass spec, or if you like increase the windows of accuracy, the hit does not actually as accurate. So you can get all different proteins as you hit, but probably false because it's basically it's a search engine. Okay. What we also can do on this machine, and that's an example also of the nanospray. You can fly the protein into the machine. And shown here is a signal for HIV dot protein, which basically is an 80 kilodalton protein, which has, in this case, 13 charges. So it's a multiple charged protein, which basically you have M to Z ratio here. So all the charges increase this number, and it makes it suitable for the analysis in the machine. But also because it's nano, and what it does, it increases the number of ions generated by the process, and it actually increases the number of the background noise significantly. So at this point, we don't want to go back to ASI, we'd like to stay with it. And so it's kind of, I guess, now we're going back in. So, unfortunately, we can't work any further with those proteins because the program which the company developed has limitations, so you can't work with multiply charged ions after they're created by for MSMS analysis, but we hope to generate our own programs to the analysis. And another example, what we did, we interested in post-translational modification. So we used short peptide which has two phosphate groups chemically attached, and we were able to apply this peptide on non-LC or non-ASI mode, degrade it using high energy collision dissociation, and then generate spectrum which was nicely aligned with the sequence of the peptide and showed us actually two phosphorylations. So it's possible actually, if you have phosphate proteins, you can actually analyze them on this machine and get the information. Right. So basically, our plan for now is so we're working on the website where we will describe information about the core facility and electronic signing, and at this point we're not busy, so we can do one-to-one training basically for the users or for anybody who's interested. They can come to us and talk and get hands-on training, or at least talk to us. Right. So in terms of education, we plan to start this seminar series, and we like to include our university students into those seminars and training courses. We'd like to include students as users of Office practice, and of course we'd like to open this to researchers. And so the conclusion: so we have personnel hired, we have major pieces of equipment installed, and will be operational in early December. Full setup will be operational. We plan to already have some samples from Howard and outside Howard. So we actually plan with people who are interested at this point to use the machine. So we plan to do experiments in December, early December, and probably by summer we will be fully operational. So.
Where is your facility located right now, and will it be there in the future, or are you planning to move?
Yeah, so we are in the hospital. It's 3C27. It's the third floor of the hospital. Right. So I think the original plan was to have it in the Adams building, So there are a few problems with that. One is you have to have special power line. So it took us 6 months basically to negotiate with the hospital to get this power line built. But it's not actually that long. It's the contractor can do in a couple of days, in fact, as soon as you decide where the line is coming from. It's not difficult. But Adam's building doesn't have this power capability. So they tried to build it up for us to boost up the power, I think, capabilities by bringing in extra transformer. So that's one problem. The second problem is you need to have, I think, sufficient air conditioning. Because in the hospital it's actually good, but even with this good air conditioning hospital, the room is hot right now. So I think Adams doesn't have to. Thinking now is that they will do the whole air conditioning system in Adams, and after that we can actually come back and they'll bring this setup for us.
Another quick question. You have LC-MS, nanospray, nano LC, but you also have two-dimensional gel capability.
Right.
Are you focusing right now on LC as an interface, or you also plan to bring the 2D gel online as well, or what's your plan with that?
Right.
I'm just curious, not that it matters.
Right, yeah, sure. Yeah, I mean, we can run 2D gels if someone is interested. My plan actually is to try to do 2D chromatography, to do basically online separation. And the way it could be done, the way I think it could be done actually offline by just doing chromatography first dimension. It's usually a pretty strong anion exchanger. Or cation exchanger, depending on your protein, and then reverse phase, which is best suited for the LCMS. Or it could be online, so we need extra valve basically. So we can use extra valve upstream of the in the machine, which has space for that. So we can do basically one-step evolution from first column into the reverse phase column, and then do breaking evolution, go to mass spec, and come back to second step evolution somehow. I think that's the most important approach. People try to get away from the gels. And the reason is that we will 90% of the material in the gel.
Yeah. Do you use any admin sequencing at all?
Here, no. No, we didn't. No. I had experience in the past. I was setting up this machine back at George Washington University. So actually there is a nice kind of relatively modern admin machine by Protein Helmer, produced by Protein Helmer. However, it takes about 1 day to run one protein, and sensitivity is not very good. Sensitivity for admin is you need to have maybe 10 picomoles, I think, of protein.
Yeah, you need more.
Maybe 100. If internalness is not low, if it's low, then it's different story. For mass spec, you need much lower. You need 10 picomoles. So it depends what you do. If you're just looking at the protein profile, it's not an atomos. You need information about the modification of this relation. I think it's pretty much the end of the story for the admin. People probably still use it, but most people switch to mass spectrometry.
Do you analyze whole proteins or is it just peptides?
Right, so the plan now is to analyze peptides. Right, and then as I showed you full protein. So at this point we can run your full protein through the machine, and it will give you like a bunch of peaks. We can deconvolute those peaks, and it will give you exactly the mass of the protein. And that's about it. I think that's all we can do. I think Thermo now talking about this top to the bottom sequencing when you run the proteins. this approach has, it has advantages, of course, but it has a lot of disadvantages. And disadvantages that it's very difficult to break protein down. Because when you have peptides, they're rigid, so you can shoot them into the helium or by other means, and they'll decompose basically or crack down. If you have cool protein, it's too little and avoid the degradation. So if you're interested in something inside the protein, you probably won't even you won't be able to do that, even if you have the best machine and best procedure, because you can only get spectrum from N-terminus and C-terminus from the protein. I mean, in principle, it's possible. And also, the problem which we found was that the program that they have right now, it's only analyzed up to like charge 7, charge 8. So if you have large protein, you have to have the 15 charges per molecule to be able to see them in mass speed. And so, right now, we don't understand how it's done. I think that's the problems probably being developed right now.